Our next speaker is Kerstin Neumann from IPK. She is group leader of the automated plant phenotyping group, which is, or the group is responsible for whole plant phenotypic infrastructures here at IPK, both the management, the operation, as well as the use. And Kerstin will present an overview about the whole plant phenotyping facilities and uses in controlled environments here at IPK. Kerstin, it's on you.
Yeah. Oh,
and I need to help you probably.
So I didn't share, right?
Right, you didn't share.
Because I see the presentation. Okay.
All right.
So I think now also the online participants can see it. Yeah, thank you very much, Thomas. And we want to now introduce you a bit into the phenotyping installations here at IPK. So phenotyping at IPK. And please interrupt me or stop me if I get too long. Of course, Thomas has already many times introduced the different phenotyping networks that we are involved in. And of course, we have more levels of plant phenotyping than just the whole plant phenotyping platforms. However, today we will focus on those, and also the guided tour will be for the phytochamber and greenhouse installations and for the IPK phenosphere, which is the new building, which you will hear about in a minute. And just as a quick reminder, of course, also these installations are open in the frame of DPPN access that Uli just has mentioned, where the deadline of application is the 20th of June. And you find all the information how to apply application deadlines and the submission platform on the corresponding website. So feel free to contact us, of course, only true for German academia. Moving to IPK installations. So we have three different platforms that we call controlled environment whole plant phenotyping platforms. These are all pod-based systems, and they're installed whether in semi-controlled climatized greenhouses or in a phytochamber, and they refer to different plant species regarding their size. So for plant species like Arabidopsis, we have the installation in the phytochamber. And for medium-sized plant species such as cereals or other in these dimensions, we can use the medium-sized platform. And for the tall plant species such as maize, we have this large installation in this large greenhouse, which you will see during the tour, or we will see if we have enough time to show also those platforms. All these platforms are basically of the same principle. So it's the plant to sensor principle. We have the conveyor belts that transport the plants to the imaging chambers, and in each of the imaging towers, or we have 3 imaging towers, and they are equipped with visible light, so RGB imaging, near infrared imaging, and static fluorescence imaging from top and from side view. This is true for all 3 systems. Moreover, they have, of course, controlled watering balance systems to enable really controlled watering setups, and in particular, of course, useful for studying drought. And the capacities are different, so we will go through that in the next slide. And we have these wireless environmental sensor networks that also record the environmental conditions, and we can also put soil sensors into that are wireless that move with the plants to monitor the soil water status. So from the RGB images, you get kind of architectural traits, proxies for biomass, also color related traits. From the static fluorescence, we have the levels of fluorophores, including the chlorophyll. And from the near infrared measurements, you can get measures related to the water content. However, also those platforms within DPPN have been upgraded, all 3 with kinetic chlorophyll fluorescence imaging, the fluorochem by PSI, and you see here an example from the medium-sized platform. So we have this top view fluorochem panel, and you also have a light adaptation tunnel in front of the imaging chamber where you can weather dark adapt or light adapt the plants, and then perform, depending on your needs, various protocols for photosynthetic measurements. Yeah, just to move a bit closer into the system for small plants located in a controlled or fully controlled phytochamber. You see here this conveyor belt, you see here the shuttle door where then plants move through the imaging chamber, which is here behind in this room. And in the imaging room, you have then the imaging towers and the watering system. Here you have a capacity when you put a single pot on each carrier of 384 plants, and you can increase this capacity up to 4,608 plants if you stay in the very early stage, and you can use this trace here of 12. Yeah, so this, what we can see, we had already covered. What also has been done within DPPN is an upgrade for parallel route phenotyping along with the shoot phenotyping. This is a standalone version, so it's not automatically integrated, but these carriers are rather small, and it's easy manually to also take them off the system, bring them to this imaging box, and then also phenotype the routes. However, you need these custom-made carriers, so you need to exchange the regular carriers, and you need to put this magenta box pots, and then you have in this imaging box the two UV sensitive cameras that image then the side and the bottom of this transparent pot, and you can see here an example of a route picture. In the medium-sized system, we have a capacity of 520 single plants, so that's actually the oldest installation here at IPK, and was one of the very first systems produced by LambdaTech at its time. Yeah, and I think we already explained all the different components. Here's just another example from this fluorochem panel, so you can, of course, adjust it also in height, and here's a look into the light adaptation tunnel. The system for large plants is shown here on this slide. As said, it was designed for maize, and you can also here alternate the capacity. If you grow with single plants, so then you would need to exchange this carrier top or lid and have a hole for a big pot, and then you can go 396 single plants, or you can put up to 4 plants per carrier and increase up to 1,584 plants per experiment. And yeah, so the dimensions, of course, with 2.30 m maximum plant heights are designed for maize. And we actually also have maize running on the system, although it's not yet in this plant stage. Also here, a look into the light adaptation tunnel and the fluorochem panel. And here you have the watering station, which then waters the 4 pots. Here within DPPN, an automated integration of route phenotyping was realized by developing these customized carriers and pots that have here this transparent NER filter front disk where then the routes are visible. And if you then use this carriers set, you can have then 396. And you can do also nicely parallel route imaging along, of course, with the shoot imaging or the photosynthetic measurements with the fluorochem. And this near sensitive camera is installed in the light adaptation tunnel. Yeah, then we come to the newest installations in the so-called IPK Phenosphere, which is this large building that you already saw on Thomas' talk, and which we will go to visit in the tour. It's located here close to the park. So very nice location. If you make a little break, you can go to the botanical garden. And this phenosphere is really designed for assessing plant productivity traits and to phenotype in designed and relevant environments. It has a cultivation area of 756 square meters. And as you can see already here on the scheme, is divided into 2 actually 4 compartments, but in 2 of these compartments you have the rhizotron system, and in the other 2 you have the container system, which is also more visible here. So this is a container system, and we will actually hear a talk on this from Mark Heuermann later on today, while the rhizotron system will be addressed by Markus afterwards. And just from the capacity point of view, so the rhizotron system offers the possibility to phenotype 360 plants or rhizotron. You could probably also put multiple plants there if you have an established image analysis for differentiating the plants. They have a depth of 90 centimeters. So you can at least, as you see here, phenotype, for example, cereals up to the flowering stage. The container platform is really designed for this field-like growth in this plant stands in these large volume growth containers, and you have the phenocrane that has the multi-sensor setup and is equipped with RGB camera, 3D laser scanner, hyperspectral camera, and this FluoCam for kinetic chlorophyll fluorescence. But we will see this, of course, then in the tour. And yeah, especially the container system is made for the simulation of field-like but controlled conditions. So you can repeat experiments, unlike in the field where you never can repeat the same field conditions, and also to address future climate scenarios. Of course, we also here have this challenge of data management, and I think we have a good team around the data management. And also, of course, IPK follows the FAIR data paradigm that wants to make the data findable, accessible, interoperable, and reusable. And we follow here the MIAP recommendations and have then define standardized metadata formats where we fill in the information about experiments, and those along with the result data and original images, for example, are then stored in our internal laboratory management system, the LIMS, which is then called the PhenoLIMS for this application. And we also have an infrastructure to publish phenomic data and to have a data repository, which is the EDAL system, and you get a DOI for your data sets, which you upload there. There's also a review process, and there have been also first data sets published with this infrastructure. This is just illustrated here, the phenomics data management and publication at IPK. So as a heart, you have the HSM system and the Oracle database and the Limsophy, and you can export data and of course import the relevant raw data that you want to store. And this is all managed by the bioinformatics group headed by Uwe Scholz, and there are Matthias, Daniel, Danuta, and Junaid, which are involved in this whole data management. Yeah, just coming in the last part of the talk to some applications of this whole plant phenotyping platform. So we used especially the medium-sized system substantially for drought tolerance studies. And just a very recent picture of the drought monitor in Germany. So this is a map of Germany. This is the middle of Germany where also IPK is located. And at least until last Friday, we had another example of a spring drought experiment. And people from Germany, we remember the years 2018, 19, and 20, where we had really 3 consecutive years of drought also here in Germany. So it's a relevant problem also for our regions. And we had started years ago with using the system for studying a barley collection, which came from the IPK gene bank. So it were single seed descended lines. All 2-row spring types, and we have established and applied a setup for spring drought conditions, where of course in the well water treatment you don't have any stress, while in the drought treatment we had this stress period during the tillering phase, which ended before the flowering stage. And we did daily imaging on watering. And then use this generated data for genome-wide association studies of biomass formation in well water conditions and under these spring drought conditions. And there were, at this time, we performed 3 experiments for the well water conditions and 3 for the drought stress conditions with 5 biological replicates per line. So that then after analyzing this data statistically, these are here the blues within the experiments from these 100 spring barley lines. In the well water treatment, you see a more or less constant growth following a logistic growth curve. You also see a bit of seasonal effect because we are in the semi-controlled greenhouse. So the second experiment running over summer had a bit more or higher biomass production. In the drought treatment, it looks like this. You have this wilting curve during the drought. Of course, plants still can grow, and once the stress gets too strong, they stop their growth, and this digital proxy of biomass, which is taken from the image from the site, so the site area and the top area, this volume equation, then this decreases, and this is basically reflecting a loss of water in these plants. Once you re-water the plants, the leaves get erect again, and you have the recovery, or can follow nicely the recovery of plants. Of course, they never reach the same biomass as in control conditions. We were also very happy at this time when we looked at the heritability. So even under these drought conditions, in these semi-controlled conditions, we have very good heritability estimates ranging from 0.59 up to 0.91 across all days and both treatments. And there's always a bit less heritability for the very early biomass when the plants grow very fast. You can also visualize the phenotypic correlations over time. Here, the example from the well water treatment. So these are now day 10, which is the first day where we could assess this digital volume or the estimated biovolume until day 58 when the experiment ended. So these are all the biomass days correlated with each other. And if you look here along the line, you see that for early biomass at day 10, you have some correlation to the adjacent days, but this gets very fast lost, and there's no correlation to the later time points, while when you look at late biomass, so day 58 correlated with all the other time points, you have a stronger correlation to the adjacent days, which is getting lost less fast. And of course, again, no correlation to the early biomass. Similar, and this, of course, we expected then due to this strong dynamic, which we realized that there would be also different genetic loci to be expected for biomass over time. And similar, it looked in the drought treatment, the drought stress period biomass was not correlated with the other treatments. So also there you would expect different genetic loci during the strong drought stress period. And in fact, this is what we see. So these were examples here of QTLs we detected. So what you see here is again the timeline and the minus log p value of the corresponding SNP to that QTL. The blue values belong to the well-watered conditions and the orange one to the drought stress conditions. And you have here an example for an early biomass QTL, which is there in both data sets. So we analyzed them independently at this time, and then they disappear in both data sets, while you have other QTLs, which are of course the most interesting, which come only up under drought stress conditions in the drought stress phase and then disappear with rewatering. So those are the drought adaptive QTL. And we had constitutive biomass QTL, which come up in both of the treatments, although not with the same importance. So here the locus on 7H was the most important biomass QTL under well-watered conditions, while under drought stress conditions in the recovery period, this was the locus around the PPDH1 flowering time gene. And we discovered in this data set only one QTL for the recovery phase, which is not corresponding with well-water treatment. So this is how you can then look at the temporal resolution of the genetic architecture of biomass. We also tried to check, so we used growth modeling. I said the logistic growth curve fits best to the well-watered conditions, while in the drought stress period, we had here this bell-shaped model, which gave us the time point of each plant when it stopped growing. We called that wilting time. And then we were interested, so is there a common water status coming from the watering data of the system where this happens in barley? And these are here the watering data in these three experiments based again on the blues within the barley panel. And then for each individual plant, of course, we checked what water status was that. So maybe first to the wilting time. So the average wilting time was at 36.9 days after sowing, with a good or high heritability and a large range within this diverse barley panel. And then we crossed for each plant the information what water status was at this particular wilting time. And also this trait has a good heritability, and the average in the collection was as 21%, but there was a large range again in the barley panel. And based on this data, we then defined this 20% water content as a threshold for severe drought stress level when you then conduct experiments which have a longer drought period, for example. And we repeated this once the fluorochem was there. We repeated this experiment and then also monitored the photosynthetic efficiency of photosystem 2, so the QY. And you see that during the well-watered phase, there's not much going on, while in the drought stress phase, photosynthesis is decreasing and recovering again. And this was also then the time when this drought adaptive biomass QTL occurred. Yeah, as said, we then also modified the setup to cover a full life cycle. So we had a collaboration with Joshua Saranda from the Hebrew University, who detected a QTL for yield under drought in the field from wild emmer. And the 2 QTLs were introgressed into Israeli whether bread wheat or durum wheat cultivars. And we were then here conducting one experiment where we had the plants from the day of sowing until day of maturity on the platform. And we simulated this drought in the field, which is developing slowly over time and intensifying in the plant ripening stage. And that worked pretty well. So we have here the example of the 7A QTL, the Nile introgressed into Barnier. And yeah, under well-watered conditions, these are here the 2 blue lines. The Nile produced already much more biomass, and that was also true under drought stress conditions. So this is here the Nile, and this is the corresponding parent. There were no differences really in the flowering time. So this is really a different regulation. And also we saw that the Nile corresponds much later to the drought treatment than the parent. Further, when you look on plant height, then it was visible that the Nile, so the Nile carrying the QTL was far taller in both treatments. And interesting was the color value coming from the RGB images. So when you compare here in the well-water treatment, yeah, there is no difference between the near-isogenic line and the parent. And in the drought treatment, they start to go to senescence earlier, and you see there's like a kind of stay green effect of the near-isogenic line compared to the parent. And this QTL was then, we also evaluated different traits, and this was connected with a larger fleck leaf area, higher photosynthetic capacity, higher final biomass, higher final grain weight, higher water use efficiency throughout the life cycle, and a higher 1,000 kernel weight. So we could really yeah, validate this yield advantage of the near-isogenic line. And yeah, for this other combination where the same QTL was introgressed into durum wheat, we also saw this stay green effect, however, here also in the well-watered conditions. And this was not exactly the same as the other combination. So here we only saw a longer peduncle and a longer last internode. We also saw the higher 1,000 kernel weight here, but here it was connected with a lower grain number per year, at least in our setup. Just for the time, I want to summarize and conclude. So we have these 3 whole plant phenotyping platforms that allow to phenotype plants of different size. It's equipped with RGB, near infrared fluorescence, and fluorochem imaging, and of course watering, and with options for parallel root phenotyping. And these systems allow to study the dynamics of growth and trait relationships throughout the life cycle. We get high quality data in these controlled environments and that allow for low replicate number in our experiments. And you can resolve temporal dynamics of trait relationships or also of genetic architecture. You can growth model curves and yeah, use different traits to come up with new traits, so trait integration. And we have then developed these thresholds for yeah, severe or mild drought stress scenarios. And of course, all these phenomics now can reveal relevant time points for sampling for this multi-omics data that then help to interpret the growth pattern of plants. And of course, this is a huge group of people who is involved in all these experiments. So of course, Thomas and Astrid for all the installations and thoughts, and all the people from the group involved in the root and shoot phenotyping, the image analysis groups, former and current members, the IT group, the quantitative genetics group here at IPK, also my former working group, and not to forget our infrastructure technical group and the experimental field and nurseries. And I hope I didn't take too much time. I thank you for your attention and I'm happy that we will have the tour to show you this in real situation.
All right, Kerstin, thank you very much for the overview. And again, the talk is open for questions. Hendrik, here in the audience.
Yeah, Kerstin, this is all wonderful what you told us. But let's go look ahead and see what in the end, we want to understand what the plants are doing. What do you feel are the 3 major bottlenecks now you still have to solve to really understand what the plants are doing?
I mean, one is, at least with the whole plant installations, of course, it's still pot-based system. So the translation of pot to what, for example, cereals that usually do in the pot a lot of tillering. They don't do this in a plot in the field, and the translation, therefore, especially regarding yield data, is usually not that great. That was this one example with the QTL where it worked well. So that's one challenge of these pot-based experiments, which will be addressed by the container system in the plant cultivation hall. Another thing is simply still also the data analysis. So you have many data, especially if you add omics. So this simply time consuming, and then the data integration and then interpretation. So it's all doable, but it's still everything time consuming before you come up with the real conclusions about the data set. You have performed many other experiments in that time that need analysis. Yeah, and of course the complexity of all these interactions. Yeah.
Thanks.
Okay, Philip. Yeah. And please, people in the Zoom meeting, raise your hands if you have questions. But Philip is next here.
Much, Kerstin. That was a very interesting talk. Of course, the technology side is quite impressive, I think. Don't have to say that, but what's really interesting is, of course, what you're doing with all that technology. And you were talking at the end of your talk about relevant time points. In that direction, I want to ask my question, and that is, it appeared a little bit like that. Did all the plants that you investigated in your GWAS experiments recover from drought, or were there some lines that had been pushed over the permanent wilting point?
In this particular setup, no. So we didn't lose a genotype. I mean, there were 5 replicates at this time, so I can't recall if there was maybe a single plant that was not recovering from the very sensitive genotypes, but at least it was no genotype completely failing. So we also had a lower level, which was 10%, I should say, where we didn't know if that will work out, but it did. And nowadays we use the 20% if we go for longer drought periods, because with the 10% over a long time, you would still risk, I think, to lose plants. Of course, needs to be established for each species. So this is our threshold for cereals. Yeah.
Particularly interesting in respect to future climate change scenarios or the general climate here in Germany, actually in face of that quite extreme spring that we are having now is, of course, and there I just want to ask your opinion. Would you say that there is value in the information on how long or how severe a drought must be in all these extents, length or severity, to push our crop species beyond the point of recovery?
If it would be interesting to phenotype that.
Exactly. Would phenotyping have
it would be interesting, of course. I don't know how heritable this trait would be. I mean, we have this wilting time or this time point where the growth stops, and we see already there large variations. So I would also think that regarding the time point when the drought is too severe to then recover would be of interest. We haven't done such experiments, but I think it's doable, probably a good idea. What I have to say, what we see in this diverse panel, when you have strong biomass differences already in the well water control, what's of course important for the drought phenotyping or for this drought related parameter wilting time is the initial biomass when the drought starts. So the high biomass genotypes, they tend to wilt earlier, of course, because they consume more water. and reach this point earlier. However, if you integrate that information, you can still detect genotypes with the same starting biomass that differ in this wilting time. I would expect that probably also for this point. No?
Okay, there is a question in the online Zoom meeting. Wai Chen, please unmute yourself and go ahead and ask your question.
Hello, Kerstin. Thanks for your nice presentation. Can you hear me?
Yes, we hear you well.
Okay, thanks. Okay, great. I noticed that you talk about the biomass that is always the digital biomass that is estimated from the image data, right?
Yeah, exactly.
So the
estimate
question is how frequently do you need to check the goodness of this digital biomass? Are they always or what is the checking strategy to match the digital biomass with the real measured biomass?
Yeah, I mean, these are not anymore done frequently, but of course, in the initial times, we have done this very much, that we have done the destructive measurements of biomass, and you then check the correlation to the digital proxy, which was in all the data sets regarding wheat and barley, where we have made this calibration very high. And we also had then a reference experiment where we had a staggered sowing and then we're bringing them to the phenotyping platform one. So you had plants of different cultivars and of different developmental stage imaged one day, and this correlation to the biomass proxy from the images was very high. So this is very reliable, and we see that in all the plant species we have looked at so far.
Thank you.
Right. Is there a further question? Either here, yeah, Henrik?
Yes, in many of, I've never done a drought experiment myself, but in many experiments which I read, I see that if there is a recovery phase, plants can enormously compensate for the losses they have had before. Did you look into any clues about which genotypes are particularly good in this recovery?
Now, we did the GWAS, so we detected also a QTL in barley. It was only one QTL. Later, we went into 220 diverse winter wheat lines, or breeding winter wheat lines, I should say, where we detected more recovery adaptive QTL. So there seems to be a genetic basis for this. We have, of course, checked another experiment where we then outside the platform grow the plants to maturity. So of course, they recover, but they can never recover to the full extent. So there's an average loss of grain yield, plant-based grain yield of 35% less than the well water treatment. And what we have seen then later adding the photosynthesis information and of course looking on the growth rates is that of course in the recovery phase, the relative growth rate is higher. And that's also the time where the photosynthesis correlates with the biomass. So not in the drought phase, the QY from the photosynthesis measurements, but in the recovery phase. So there seems to be a high importance of photosynthesis because it might be a limiting factor in this period.
And other physiological changes that the plant can do to adjust either to the drought phase or to the recovery phase? Have you looked into that?
Yeah, we have. I mean, we didn't make measurements really of other physiological traits despite the photosynthesis measurements, but we see also an increase in the water use efficiency. But this is also, I think, not new. We are at the moment adding omics data, but we don't have the data yet. So we have done experiments with Roberto Tuberosa in the EPPN experiments where you had this durum wheat or selected lines from the durum wheat GWAS panel for osmotic adjustment on the system. And we have really nicely before said we will take at this water status samples for metabolomics, and they are already so they are already processed, but the data are being analyzed right now. So I can't yet report on other mechanisms.
Stop recording now. Oh, there is another question.
If you could get everything you wish for, what would be the trait that you would wish to have, but which you can't currently measure? Are there other things which you see in your data set? It would be really great to get an insight into that, but we can't technically do it, for example, at the moment, or we don't know how to analyze the data to get it. What would be your opinion on the sorts of things that we should aim towards?
Oof, I think there could be a high list. What comes to my mind, for example, are the cuticular leaf surfaces, how many stomata you have, are they open or not? So such things you can't yet really see from phenotyping, at least not in these high throughput systems. So that would be great to add during these experiments. But there might be many others that now don't come to my mind. But good question, of course.